Okay, good evening and half a day, everybody. Welcome to the University of Guam Presidential Lecture Series featuring Dr. Matthew Farr. My name is Jonas Macapinlac. I'm the Director of Integrated Marketing Communications here at the University of Guam, and I will be your Master of Ceremonies for the evening. At this time, I'd like to recognize some of our distinguished guests in the audience. First of all, Dr. Robert Underwood, 10th President of the University of Guam. Dr. John Peterson, Assistant Vice President, Graduate Studies, Sponsored Programs and Research. Mr. Troy McVeigh, Associate Dean, College of Liberal Arts and Social Sciences. The Honorable Benjamin J.F. Cruz, Vice Speaker, 32nd Guam Legislature. The Honorable Frank B. Uggan Jr., Senator, 32nd Guam Legislature. I'd also like to recognize Mr. Kwang Ming Chen, Commissioner of the Overseas Community Affairs Council, the Republic of China. And now please give a warm welcome to the 10th President of the University of Guam, Dr. Robert Underwood.
Today, thank you, Jonas, and we look forward to this presentation by Dr. Matthew Farr. He comes to us from the United Kingdom via Canada. He's a professor at the Department of Medical Genetics, University of British Columbia, and he is a Canada Excellence Research Chair, which is a very prestigious title. There's only 25 of them in Canada, in Neurogenetics and Translational Neurosciences, and he will be our 23rd speaker in this lecture series. His presentation is entitled Parkinson's Disease, Ontology and Etiology. He brings to us, he's the second most quoted researcher on Parkinson's disease in the world today. Give him a few more years and I'm sure he'll be number one. Matt has published more than 350 papers on Parkinson's disease and movement disorders during a career spanning 17 years in Science and Nature and New England Journal of Medicine. He has active collaborations in human genetics and neurology in more than 20 countries around the world. And of course his work has attracted a great deal of media attention, including a 30-minute PBS frontline documentary entitled My Father, My Brother, and Me. He also is the younger brother of one of our faculty members here, Professor of Anthropology, Dr. Douglas Farr. So, you know, he's just a kid brother. And I know how that feels. You know, as a kid brother to somebody all my life. Matt brings to us a wealth of experience and a wealth of knowledge. And I can tell by the people that are here this evening that we have a number of physicians, we have a number of people who are interested in this, have had a long-term interest in this. We also have family members, I know, of people who have been affected by this disease. So everyone is very interested in. He gave me one insight, which I'd like to share to you, that the quickest way to somebody's neurology, somebody's brain, is through their genes. And so who knew? But he knows. And so I present to you Dr. Matt Farr.
I feel like I'm in the jungle. But it's a delight, thank you. That was a very kind introduction. I should first of all like to say why I'm here really. I'm on vacation, to be honest. I'm here seeing my brother and his family, who I haven't caught up with in many years. His daughters are now 12 and 9, and I've I think I've seen only one of them once when she was 1 or 2 years of age. So that's pretty shocking, really. But I've got an excuse. I've been pretty busy, and I've been very passionate about Parkinson's disease for many years, and I remain very passionate about it, and trying to solve a puzzle, a riddle, of why this disease occurs, and what we can do to slow it down or to halt its progression, and using the tools that I know best, which is genetics. So it's a rather strange way of coming to study the disease. I'm not a clinician, I'm not a neurologist. I can't advise people on how much cinema they should be taking, etc. But I want to tell you a little bit about the molecular revolution that's happening in the world in medicine, and it's happening today through genome sequencing. This is the building where I now work, and I'm on the 5th floor here. It's a new purpose-built facility, and it integrates neurology, neuropsychiatry, neurosciences, neuroimaging, and neurogenetics. It's the 5 big N's, if you like, all together in one building. It's a multidisciplinary team of people all working together to solve problems in neurology and neuropsychiatry through modern means. And one of the things I'm building is a genome draft for brain health, to try and understand how genetic variability contributes to brain health and to brain disease. I do that because I'm a geneticist, I map genes for disease, and I've been doing it for some years quite successfully in Parkinson's. I'm going to tell you a bit about some of those discoveries. This isn't for the faint of heart, so hang on to your chairs because it's going to be quite fast and furious. It's very difficult to give these types of talks because some people in the audience are very professional, very knowledgeable about, say, clinical neurology. Some people in the audience can be very professional and very knowledgeable about maybe population genetics. Some people in the audience know the disease better than anybody else because they have it, or they have a family member with it. And to try and address all of those people and make them all happy and give them something to take home and a message of hope is a challenge. So forgive me, and up front, I'm going to do my best to try and not make it too esoteric. So what I do is these different things. I try and integrate in my own little way information from neurology and neuropathology. So for instance, clinical diagnoses of Parkinson's disease, pathologic diagnoses of Parkinson's disease. And I work very closely with populations around the world. I'm trained as a population geneticist, human geneticist, originally a biochemist, and then morphed into neuroscience. And I work with lots of populations around the world and families within those populations, and many of them are isolates. So the people living on islands like Guam, or the Faroe Islands, or Tunisia, and I'm going to tell you about some of these countries, or remote villages in northern Norway, and families from these isolates as well, so even more homogeneous, if you like, in terms of genetic background. I'm going to take you through some of these family studies, and what we do is we map mutations underlying the susceptibility for neurologic diseases, mostly Parkinson's disease. And when we find those gene mutations, I generally model them in mice by making conditional knock-in mutant mice, and then to try and develop assays that are relevant to the human condition, but in a dish, that we can work with the pharmaceutical industry to try and address the problem. So most medications for Parkinson's, I'm going to take you through it, are for symptomatic benefit. They make you feel better for a limited period of time, but they don't slow down the disease's progression, unfortunately. And my ambition is to slow down the disease's progression as a means of therapy. And we take this all the way back through imaging and through biomarkers. So I'm going to give you illustrations of this through the talk. First of all, for those of you who don't know, Parkinson's disease is considered a movement disorder. It's the second most prevalent age-associated neurodegenerative movement disorder in the world. It knows no territorial or country divides. It basically affects all populations in the world similarly at relatively similar frequencies. It's progressive, and subjects with it generally suffer an inexorable an unstoppable decline in their disease. Pathologically, it's pathologically it's noted because you get a loss of black substance, of substantia nigra. So this is the midbrain and the brainstem, part of the brain, getting section through that, of a normal individual. And normally this is black substance, substantia nigra, black substance. And in Parkinson's patients, this is gone. This region of black pigmented neurons is something in the order of 700,000 neurons. And in patients with Parkinson's disease, you generally have maybe about 300,000 neurons. So they've lost about 350, 400,000 neurons to start to manifest symptoms of the disease. And the loss is progressive. It was first described in 1817 by James Parkinson, but it was actually probably described in Ayurvedas in India 5,000 years before that. So it's been around in the medical literature for a very, very long time. James did a very good job of writing down an essay. It's basically a description of only 5 patients, but he got it right. He noticed a lot of different features of the disease that kind of fallen by the wayside. So Parkinson's disease is not just a movement disorder. There's a lot of other, say, neuropsychiatric components. There's problems with gait and balance, leading to lots of falls and injuries. There's problems with sleep disorders. There's problems with autonomic disorders as well. So it's a syndrome, a multifaceted syndrome. And many of my neurology colleagues that I talk to tell me that Parkinson's disease is kind of unique. Every patient that they've seen or met with Parkinson's disease has an almost unique constellation of these features in a sense, in terms of their progression and in terms of which ones bother those subjects most. In terms of so Parkinson's disease is a bit of an umbrella term, or Parkinsonism, for a whole variety of different disorders. Parkinson's disease being the most common is associated with a triad of stiffness and slowness and tremor and possibly postural instability. Generally starts asymmetrically down one side of the body, and patients do fairly well on levodopa replacement therapy. So replacing the dopamine neurotransmitter that those neurons in the brain normally produce. The median age or mean age of onset is around 70 years of age. But there are a whole host of other forms of Parkinsonism, and I've separated them out here pathologically into synucleinopathies, tauopathies, and others. And you might notice one here, Lysicopodia of Guam, and I'll mention a little bit about that at the end of the talk, because I've done a little work on that prior to coming to Guam, knowing that I'm going to visit my brother and knowing that I'm going to visit John Steele in the audience, so I thought that would be good to do. In terms of genetics, when I got into this field, everyone said to me, "You're an idiot. You're a geneticist, go and work on something genetic." Parkinson's disease is a sporadic condition, it has no genetic etiology, it doesn't travel down the family line for the most part. It's extremely rare when it does, and if you read any of the older medical textbooks, you'll find that that's the description of PD. It's not a genetic disorder. But everything that lives, moves, breathes, walks, whatever, on the planet has a genetic blueprint, and therefore has genetic determinants that affect how that organism behaves in its environment, affects its susceptibility to disease, for example. And so that's pretty much what we're talking about. Age is probably the biggest risk factor for Parkinson's disease. You still have to get to 70 years of age for the most part to manifest signs and symptoms. And even in the families where we've mapped single nucleotide changes that are causal, quote unquote, you still have to get to a good age before you probably manifest symptoms of the disease. So please don't take this as being genetic determinism. It's not. It's really as a means to an end, to find out what is really going wrong at a molecular level in this disorder. What are the parts of the engine, if you like, in that car, and how do they fit together? How do they work together to make that motor run? So I was told I was a fool to get into genetics, but in the last 17 or 18 years that I've been working on the condition, I've managed to develop collaborations in many, many countries around the world. Because, like I said, Parkinson's knows no boundaries. And I'm going to talk to you today a little bit about these genes here that have had a direct role in mapping, and a little bit about their function, a little bit how they work together to basically provide a pathway, if you like, that's affected in garden variety Parkinson's disease, and how they're providing now major targets for the pharmaceutical industry to start developing drugs to fix the problem. And that's what's most important to me. There are many, many forms of Parkinsonism too. So this is not necessarily, for instance, these forms here are early onset forms of Parkinsonism, under the age of 45. These are quite atypical forms of Parkinsonism. I'm not really going to talk about those so much. I'm going to save it for the garden variety type of idiopathic Parkinson's disease for the most part. So I mentioned there's a lot of genes, and many people have looked at monogenic genes like these that basically find specific mutations in a specific gene, and they appear to travel down the family line. There's also studies that have been done on many thousands and thousands of patients with Parkinson's disease versus many, many thousands of controls. The last study that data that I'm aware of involved 120,000 subjects. So it's very large studies. They're international. They tend to span North America and Europe or Asia or both. And this is what we've found so far. This is a Manhattan plot because the skyline here. These are chromosomes from 1 to 22. And this is minus log 10 of the p-value, so basically it's statistical significance. And whenever you see these spikes, these are statistically significant associations with disease. And there's one here on chromosome 4, big one at 17, but there's many skyscrapers, if you like, on the Manhattan plot. And each one of these is a gene signature for Parkinson's disease. So it shows you that there is definitely a genetic etiology in idiopathic garden variety Parkinson's disease, regardless of whether your father had it or mother had it or there's anyone else in your family with it. There's a genetic etiology in Parkinson's disease. It's probably something in the order of around 27% now of genetic variance has been mapped. I'm going to talk a little bit about synuclein here and tau a little bit as we go through the talk, because these genes have also been identified through not just association, but also through a technique called linkage, which is, I think, far more powerful. So the first gene was mapped in 1997, just as I really got started. And this is the gene. It's a protein. It encodes a protein known as alpha-synuclein. It's quite small, 140 amino acids. And since then, I mean, the most recent mutations that have come out and have been published are these ones here, G51D, and H50Q. This was by my group just last year. Antibodies to the protein, so making reagents that will stick to the protein, have turned out to be the best immunological stain of the disease process in the brain to date so far. So this is a picture, an H&E staining of a brain, or sections of a brain of a patient with Parkinson's disease. And one of the things about Parkinson's disease, or most forms of Parkinson's disease, is this hallmark pathology. So this is a pigmented neuron in the substantia nigra, the black substance here, the neuromelanin. And this is what's known as a Levy body or Lewy body inclusion. It's a protein aggregate. And patients with Parkinson's disease tend to accumulate these, and also accumulate these Lewy neuritic processes here. And this is what you see in this picture. This is abnormal, it shouldn't be there. With alpha-synuclein staining, these are the same slides. So just to give you some idea of the amount of occult pathology that wasn't observed before, this gene was actually found. This pathology is enormous, and the burden of pathology in this individual's brain is extremely high. So now Bracken colleagues, and there was a very nice paper last year in Nature Reviews Neurology, to stage the disorder. And his thesis is that for idiopathic Parkinson's disease, it probably starts in the gut, in the myoteric plexus and the gut's nervous system, and ascends through the vagus, through the dorsal motor nucleus, and through the brainstem, and into the midbrain, and then into the cortex, and through this back staging scheme, which is a very nice, I think, illustration of the disease and the disease process in many, many patients, but not in all. It suggests that, I don't know, not everybody in the world gets Parkinson's disease, right? So there must be some, so what I'm trying to look for and find is genetic variability as a clue as to why some people are more likely to get it than others. There may be lots of things that are exposed because of PDE, environmental triggers, some infectious agent, some toxin they ate, many things, but those are very hard things to pin down. And I've always said to my colleagues, there's only 6 billion nucleotides in the human genome, right? 3 billion pairs, and we know what they are now. We've known for the last 10 years, we've known the exact sequence of A's, G's, C's, and T's. And so we know when one isn't right. And so we can pin this down. We can pin this down unequivocally. This is the first family, one of the first families I worked on, worked with, and on, sample from the family. This is known as the Iowa Kindred, and this is an old sepia photograph that they gave me, very kindly gave me to share. And this lady here has Parkinson's disease. She has a masked face and a dystonic hand, the way she's holding the infant here. It's a rather unusual posture to have. And all of her sons went on to get relatively early onset Parkinson's disease in their 40s. We described in Science in 2003 a triplication of a piece of the genome that contains the alpha-synuclein gene in these family members. I'm going to introduce you to Myra. I don't know if I can do this on this. You might have to help me with this, maybe. Can I make it play? I can do it on the computer. I can come to you. Just need a mouse on there. There you go. Just tap on that thing. So Myra allowed me to use this video that is taken by Katrina Gwynn. And Myra is a member of the Iowa Kindred. She's got some dyskinesias. The movement in her legs is due to the drugs that she's taking to actually help with her symptoms. And this is a side effect of those drugs. She's being asked to do rapid alternating movements like this with both hands. It's a rather quick video, but it just gives you some idea of her level of disability. This is when she's on medication. This is as good as it gets in Myra. And she's deceased now. She was a bank teller. She got the disease at around 34, 35 years of age. She had a very early onset, very aggressive form of the disease, and she died in her mid-40s. Hope they'll go forward. Can you do next slide, page down, please? That's it? Okay, that'll be it. I'll go back on there. So what we found is by looking at the genetic material from many family members, and I had to trace them down, they all had run away from the farm in Iowa because they were running away from the bug. They called it the bug in the family, and they all run away to different parts of the country. And I spent quite a long time, over a couple of years, tracking down different members of the family and asking them if they'd be willing to participate in the study. And sure enough, we did get enough blood samples to do the study. And we found this synuclein multiplication. And this is a region of chromosome 4. The piece in green here is basically the amount of DNA you should have in a neurologically healthy individual. And in these individuals from the Iowa family, they basically have twice as much DNA for this segment of the genome, for the alpha-synuclein gene. And in fact, actually, they had 4 copies rather than 2 copies that you and I have. We've done many other studies on many other families, with many other families over the years, and now find that the amount of alpha-synuclein you produce is actually associated with, or directly correlates with the age of onset of disease and the progression of that disease, and the comorbidities of that disease that subject will eventually manifest. This is a study that we did in Sweden. I was very fortunate. I basically got a DNA sample from this fellow here from the Parkinson's Institute sent to me, and this lady here from Germany sent to me by Tom Gaza, and this one's from Bill Langston. And they said to me, Matt, we don't know what's going on in these families. Can you have a look at these particular DNA samples and tell us? And so sure enough, we did some genetic screening and testing and some research. And one thing I noticed about these 2 samples is that not only did they have a synuclein multiplication, they had the same identical genetic fingerprint in their region of chromosome 4, which told me they had to be related. So I was very fortunate because I was doing a lot of work in Sweden and Scandinavia and Norway at the time. I knew about Henry Muniz' work, and this is his thesis from the 1940s, and I knew about the Lister family complex. So it turns out that these individuals were actually part of it. This is a very, very large family that is in this peninsula Lister, where there are many, many, many people with Parkinsonism and dementia living today. And the disease is what we call autosomal dominant. It passes down the generation in this family. It goes 50% of every generation is affected, and 50% of each generation thereon is affected. And what's shown here in these pictures is just this individual had 4 copies of the alpha-synuclein gene. You can actually count them. This is interphase fish of a leukocyte to a blood cell, and we've actually labeled up the alpha-synuclein piece of genomic locus with a fluorescent probe, and you can actually count they've got 4 copies. We should have 2. He got the disease at 31 years of age and had a relatively rapid decline. This lady here, she was diagnosed with a form of Parkinson's known as multiple system atrophy at 70 years of age, much later. And she has 3 copies of the alpha-synuclein gene. We have this type of picture from many of the affected family members now enlisted, and this is being followed up at the moment. So just to give you some idea, if you've inherited a synuclein, 4 copies of the alpha-synuclein gene, it seems like a misnomer, triplication. It's triplicated on the mutated allele. But if you inherit 4 copies of the alpha-synuclein gene, then your probability of being affected at the age of 50 is something in the order of 80%. You're very likely to be affected. But there are many now synuclein triplications, duplications, as well as point mutations, and this is a big area of interest. The pharma. So what does synuclein really do? Synuclein's got a role in synaptic plasticity, and it was first mapped in zebra finches, in somnolent learning in zebra finches. It helps regulate synaptic exocytosis. Tom Sudoff, who's done a lot of work in this area, looking at synaptic vesicle proteins and their dynamics, got the Nobel Prize this year, and in part because of his work on synuclein. This is synuclein here. If you knock it out its homologs, alpha, beta, and gamma synuclein in a mouse, you do get some deficits in conduction velocity and axons. You get some neurodegeneration actually in those animals. But you have to knock out all 3 copies, alpha, beta, and gamma synuclein. If you knock out just alpha synuclein, you have no effect in mice. So this has led to some very interesting thoughts about how we actually might be able to treat the condition, treat Parkinson's disease in most patients. So most patients have alpha-synucleinopathy. Most patients have Lewy body pathology. You can actually induce Lewy body pathology like this in mouse models now by injecting synuclein fibrils into the brain of an animal, like a mouse or a rodent, a rat, and within a very short period of time, you see this pathology spread, this tsunami of pathology spread throughout the rodent's brain. Nobody quite knows why, nobody knows the mechanism of transmission. It seems almost like an infectious agent, almost a prion-like process. But some people obviously are more susceptible to this process than others, perhaps, and that's why perhaps they get Parkinson's disease and perhaps dementia with Lewy bodies too, is a difference in the growth. Came to the idea that lowering synuclein therapy might be a good idea. Farah and Miller got the Nobel in 2005 for RNA interference, and so we started working on this in 2003, and the technique was out then and well established, and doing it in experiments in mouse brains and basically knocking down synuclein expression in the hippocampus. And you can see it's a pretty good dose response here. You can actually, by infusing in synuclein antisense oligomers, you can basically lower endogenous levels of neurine synuclein in the brain of an animal for several weeks, and it's a pretty good washout, pretty good characteristics for a drug for human patients. We did the same studies in non-human primates in 2010. It's probably going to be a bit technical, but similar results. And these are treating primates with MPTP and treating one side of the brain of the nigra with RNAi, alpha-synuclein, and one side of the brain with sham. My thoughts on it around this a couple-fold. There are now 2 or 3 very large companies in the world, pharmaceutical companies, actively working to develop RNAi as a therapeutic for idiopathic Parkinson's disease. I won't mention the names because it's conflict of interest, but they're very actively working on this area. And you might think, so, well, how are you going to get this drug into the brain? Well, a quite common therapy for Parkinson's disease these days is deep brain stimulation, where you put an electrode into the subthalamic nucleus and basically quieten down the signal, the output pathway, if you like, from this striatum. At the same time, this therapy, DBS, really has great symptomatic benefit, but it doesn't actually slow down the disease progression. So if you put a cannula in there at the same time and allow the drug, such as the synuclein RNAi, to be infused into the brain, ASOs especially, you could basically prevent or stop the Lewy pathology that those individuals have or will manifest and develop over time from development. And so basically it's a new way of treating the disease, and not just treating the symptoms, but treating the underlying etiology. So that's my first illustration of how molecular genetics can lead to new therapies. we went on to find this gene, leucine-rich repeat kinase 2, and I wanted to tell you a bit about this story. Most recent pathologies that have been documented are from a family known as the Sagamihara family in Japan. 6 family members have come to autopsies. They don't actually have alpha-synuclein pathology, they have tau pathology. All 6 family members have tau pathology. And LRRK2 is kind of the Rosetta Stone of Parkinsonism. It unites many of the different pathologies together like the Rosetta Stone, and of course you allowed us to decipher hieroglyphics. So I think LRRK will help us decipher the role of synuclein, ubiquitin, tau, and actually TD43 pathologies in Parkinson's disease. We mapped the disease gene in 2004, and this family, known as family D, again, it has autosomal dominant Parkinson's disease. Each generation affected, 50% of each generation affected, and we realized there's a piece of chromosome 12, Q12, segregating the disease. Realized just a few years ago that LRRK2 is a really big player in idiopathic Parkinson's disease. So it's not just the odd rare family. This study involves something like 15,000 subjects across the world, many different groups. And I wanted to point out this particular mutation. It's a susceptibility variant. It's found in 8% of patients with Parkinson's disease throughout Asia. So we're talking Korea, Taiwan, Singapore, throughout Asia, have this G2385R variant in LRRK2. It's a major risk factor. This is some work we did with Ru Mei Wu in Taipei, in the National Hospital, published a few years ago. But I really want to focus on this variant, G2019S, and the reason why is because now it's considered one of the best, most likely druggable targets for Parkinson's disease in the world. And 8 of the world's biggest pharma companies are actively engaged on multi-million-dollar programs, billion-dollar programs now to develop therapeutics around LRRK2. We mapped the gene mutation I did with a friend of mine, Jan Astley. He's the chair of neurology in Trondheim, which is up here in kind of central northern Norway. And when he's not fishing for genes with me, we're fishing for trout in Norway. He's a great guy. We found, I've been working with him for some years, since '99, and on these families with, it seemed to have there was a higher prevalence of Parkinsonism in the villages in the north here. And we didn't know why, and so he basically was very diligent about the care of these individuals, and he said to me, Matt, will you help me find out why? And so we did, and sure enough, sequencing LRRK2, it turned out that they actually have the G at this position, the glycine, position 2019 was mutated to a serine. And if you look at this sequence, it may not be intuitive, but it is to me. At the beginning, they have a DYG, a DYG, DFG, DFG. At the end, it's APE, APE. This is the activation segment of a kinase. This is what it looks like in terms of molecular modeling. This may be a very esoteric world for so many people. I'm sorry. Hopefully it's not. You're like, stop now. It's information over there. But anyway, hopefully it's okay. To try and tell you a bit more about the mutation is here, and it's the hinge, if you like, of this door. This purple door, which normally blocks this pore within the protein. This is the catalytic site of the protein, it's the engine, it's the bit that adds the phosphate group to a substrate that comes in here. And normally this doorway is blocked by this ribbon of protein. But with the G2019S mutation, the hinge is broken, and the door is always ajar. So the thought is, could we make a competitive kinase inhibitor that basically blocks the doorway as effectively as having a door without the broken hinge? And so there's been a lot of work on that over the years. But to get there and to persuade pharma to get invested, to admit to you, it normally takes between $5 and $7 billion to make a new drug for any disease. It's a big, 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 big investment. So you really have to lower the bar a lot before they jump in and say, so that's my job, I see, is to lower the bar for pharma. So we identified this mutation in Norway and in Europe. And one of the things I noticed that was very interesting about the mutation in individuals with the carriers with the mutation is that they had the same common haplotype. And by that I mean, it wasn't just the G2019S itself that they all had in common. The same region of chromosome 12 all around it was identical in all these individuals. Whether they're living in Norway, in these villages, or in Poland, or in the United States, or Ireland, we had some of these families from different countries around the world, from European countries in North America, including this first study. What was soon noted shortly afterwards that published in the New England Journal of Medicine was that 14% of all Ashkenazi Jews with Parkinson's disease have this mutation. And then also that year in 2006, the 30%, so 1 in 3 patients, Berbers, in North Africa, so along the coast here of North Africa, all the way from Morocco to Egypt, is predominantly Berber. 1 in 3 patients seen in those clinics has a G2019S mutation, and it's driving their disease susceptibility. Nobody even appreciated that disease susceptibility to Parkinson's disease was greater in this part of the world, but it's a huge social healthcare burden. This is my friend Faisal Hentati. He's the chair of neurology in Tunis, in Carthage. And this is Rachel Gibson. She was the head of she's a clinical director at GlaxoSmithKline. It's taken back in 2005 when we were doing some of these studies. One of the things I wanted to point out here was the trading routes. This is a map of the seafaring routes of the Phoenicians 1,000 years before Christ. And basically, this is how the mutation, I think, got up to Norway. It's because the Norwegians came down here, they had settlements in Carthage. We found there's been Norwegian runes found in Carthage. And sure enough, while the Norwegian Norsemen are down here trading, they also managed to take some of the gene pool back with them to Norway, and including the G2019S mutation. And I think it's probably a similar story with Tyre and with the Middle East there. The penitence of G2019S, again, is not deterministic. In Norway, if you are a carrier of this mutation, the probability that at 70 years of age that you'll be affected is around 50%. But in Tunisia, the probability at 70 years of age, if you're a carrier of this mutation, is something like 80-90%, much, much higher. So genetic background, or maybe it's environmental background, plays a big part in this. And so we wanted to get a better answer to this. If you look at idiopathic Parkinson's disease, the age-associated cumulative incidence in Norway and Tunisia is identical. Those curves overlap. So right now I'm working with 258 families in Tunis and around Tunis in North Africa, following about 1,000 G2019S carriers and going to visit them periodically. I go about every year and go and visit. There's a huge team there, of course, working with those families. And my job has been to try and find money to invest to help the Faisal team develop infrastructure to do genetic testing, genetic counseling, and to understand really why this disease to understand what this disease really is. It's a Parkinson's disease. It looks like just like garden variety Parkinson's disease, but there are some subtle differences, and there's some very interesting ones. One is the women who are carriers get the disease earlier. This study has just been replicated in the Italian population, about 5 years younger. Another is this, and for any neurology colleagues in the audience, this might be particularly interesting. Generally, people with young onset Parkinsonism, as a rule of thumb, have a relatively mild course, and it's a fairly kind of gradual progression of the Parkinsonism, and it's more mild. It doesn't have the cognitive problems so much. This is a rule of thumb, just generally. Whereas if you get the disease late in life, it's more likely you're going to have more problems, perhaps more cognitive problems, mild cognitive impairment, more dementia perhaps, more falls. So your disease is more severe. And you can separate this out quite easily between mild, moderate, and severe forms for idiopathic Parkinson's disease, seems familiar. But if you've got G2019S mutation, you can't. They're all the same. So even though you may develop the disease at 20 or at 70, your disease course is uniform, it's homogeneous. And if you're a drug company looking to try out a new medication, that's what you want, is homogeneity, because you can reduce the number of subjects you have in a clinical trial. You do the clinical trial at lower cost, you lower the bar, right? You get them invested. We know what LRRK2 does now. It's basically involved in the regulation of actin and actin polymerization and moving things around the cell. I'm not going to go into too much detail. We've made lots of mouse models. The mice get tauopathy. All of the LRRK2 mouse expression models get a tauopathy. It's a phosphorylated form of tau, and they also get dopaminergic deficit. And the knockout mice get problems with autophagy. So problems in the kidneys, you can see here with aging. If you knock out the gene, kidneys become discolored and become enlarged, and there's a problem with autophagy. They also have a problem with this this is normal wild-type mice, this is knockout mice. They don't explore the space, they're more apathetic, they're less motivated. It's kind of interesting. We've made a lot of progress with this, and we're developing conditional knock-in G2019S mouse models. And I want to try just to illustrate this, the complexity to you. we have, I mentioned in the beginning, 700,000 ligand neurons. Each one of those projects an axon, a firing axon to the striatum, this region of the brain here. Each one of those axons basically ends up in around 450,000 terminal synapses and spans maybe 5 to 7% of the striatum. And so this is what we're doing, this is complexity. Within the striatum there are different cell types, different pathways, mostly medium spiny neurons, but their firing is controlled by this structure, if you like, of cortical and thalamic stimulation coming in, glutamate stimulation coming in, and dopaminergic inhibitory neurotransmitter coming in to control the output pathway. And this is what's ruined in Parkinson's disease, the control of this neurotransmission. So now we've got a model in mice where we can look at the integration of cortical, thalamic, and striatal and nidal signals in the context of a G2019S mutation. It's very powerful. And we've been doing this, and we've been looking at, for example, DART32. I won't get into too much, but some people in your list will know DART32 is the master protein that integrates these signals. Paul Greengard got the Nobel for his work on DART about 15 years ago. I'm not going to go into too much detail, but one thing we noticed about the mice is if you grow neurons in the brains of those mice, they do it themselves. We did it. This is newly born neurons using a green fluorescent protein tracking marker. In G2019S background, those neurons don't branch. Their arbor is a lot more restricted. The number of spines they have, so each of these mushroom-shaped spines is the connection to another neuron. And in G2019S, the number of spines is far reduced. So how can we remedy this and correct the phenotype? It comes out of drugs. So huge efforts are now going into drug development to try and do that and apply that to these types of assays to put it right, competitive kinase inhibitors. Getting into the end of the talk now, these genes were found through a very arduous task of basically, well, starting off working with several families, many families around the world, and basically collecting very detailed clinical notes and pathologies and blood samples, and really it's only with the help of those families that any of it would be possible at all, because without that there'd be nothing. Right? So it's with their engagement, but it's really taken a lot of engagement over many years to do it. But then the genetics has also taken a lot of work, a huge amount of work. Because it wasn't so easy to sequence the genome 10 years ago or 5 years ago. Now it's easy. Now I can sequence 8 genomes a day. And that's what we're doing in our lab. So this is an example of that. It's called exome sequencing. It's just sequencing 1% of the coding genome. And this is the type of family that we can apply it to. So basically where we've got, in this illustration, you've got the grandparents here, the parents there. This is the present generation, and the squares are males and circles are females, and these individuals with the blackened symbols have Parkinson's disease by illustration. And by sequencing this fellow here, this lady here, and this guy here, and comparing their genomes, and looking at the intersection of the variants they share and the rarity, and looking at the substitutions that they create, we can narrow down the genetic variability in a small pedigree like this to maybe just 20 variants that may be driving disease susceptibility. It looks too good to be true, doesn't it, maybe? But it is true. This is work we did on the Swiss kindred. Just sequencing this lady and this fellow here, the arrow's moving, I'm afraid, to start with, and comparing their genomes. And basically we went down from something like 34,000 variants, 29,000 variants, whatever, in these two subjects, going down the list of different filters to getting it down to just 7 variants that these individuals share that were likely to be causing their disease, and seeing if those mutations transmitted through the family line in these families. Sure enough, we found one that did, and we found it in families from Yemen, of all places. We have good collaborations in Yemen, and in Tunisia and in the US, and it turned out that the segregation of disease was very high, chromosome 16. This is a new pathway for Parkinson's disease. It was published in 2011. It's a system known as the retromere, and BPS35, vacuolar protein sorting 35, is mutated and basically affects cargo trafficking, membrane-associated cargo trafficking within the cell, within the neuron. Most of it affects receptor recycling back to the plasma membrane, but there's also a retromirror involvement in recycling from the endosome to the Translational network. It's also from the lysosomes to multivesicular bodies to the tentacle. Anyway, so this mutation basically is perturbed in these families. We don't know why. We don't know we know the beginning, we know the etiology, and we kind of know, in some senses, the ontology, because we know that this is Parkinson's disease in these families. But we don't know all of the molecular process in between. What is it trafficking or mistrafficking? What's going wrong? How can we intervene to correct that process? And I want to give you some recent data on this. This is what it's doing. I don't know if I can show these. Can you help me? It's the mouse. If you can do the top one on the left, just click on that one. This is trafficking single particles of BPS35 in a dendrite. And you can see how fast they go. You want to click on the other one beside it. And this is with the it should work. Yeah. This is with the mutation. So we've done nothing fancy here. These are just primary neuronal cultures from mice models that have either got the mutation or don't have the mutation, where we're comparing the rates of trafficking in the dendrites of those neurons in a dish. And without the mutation, there's trafficking going on very, very fast. With the mutation, it's basically just oscillating in one place. So can we fix it? I think we can fix it. I won't go into all this, but the trafficking that it's involved is in glutamate receptors, or glutamate receptor subunits, as well as dopamine D2 receptor subunits. This is another example. This trafficking is a very local phenomenon. It's also happening within spines, which are the connections between neurons. Move on. But very recently, last month, in April, in Nature Chemical Biology, someone has developed, it's got basically pharmacological chaperone of retromirror BPS35, which stabilizes it and allows it to do what it's meant to be doing. And this isn't applied to the DN mutation, but it's just applied to retromirror in general. So one thing I didn't mention is that the retromirror is also downregulated, not just in Parkinson's disease, but also in Alzheimer's disease. And the retromirror is involved in APP processing and helps regulate the amount of A beta 4042. A bit technical, but basically this is a way, using chemical chaperones, of rescuing that APP processing, which is a new pathway, a novel way of intervening not just in PD trouble, but in Alzheimer's. Another story. This is Mennonites. So I moved to Canada 4 years ago, and I thought to myself, I've got all these Canadian federal dollars, right? They don't want me to spend it on far-flung corners of the world, like Tunisia or Taiwan. They want me to spend it in Canada, the voting public, right? And I'm sensitive to this, so I thought, well, let's find a nicer than Canada. And if you've been to Canada, it's a very heterogeneous place. People come from all over the world. And some of the people that came there in the turn of the last century, before that, in the turn of the century before, were Mennonite communities, so that suffered religious persecution in Russia and in Germany, and came to set up farms and on the prairies in the center of Canada. And I was privileged enough to meet this family, many members of them. All of these boys, basically, this is a picture from the 1920s, got Parkinson's disease. They're shown in this generation here. And it's a family that's studied by Ali Rajput and colleagues who worked with the family over many, many years. They put it down to pesticides, too much pesticides on the farm that they were using, but everybody in the family basically got the disease. And it turns out, and they have everybody Parkinson's disease, it turns out they have a, long story short, they have an N855S mutation in a gene known as DNAJC13, which, kind of long story short, basically is in exactly the same pathway as BPS35, and as LERK2. I didn't tell you, but LERK2 also co-IPs with BPS35. Many of these genes now coalesce around this central pathway. So there's BPS35, one of the genes I told you about, LERK2, I told you about that in the Tunisians. Didn't mention this one, but another day. And Remy8, or DNAJC13, its other name is Remy8. Excuse me, they all regulate what's known as the Wiskott-Aldrich scar protein homolog, big name. Basically, it's a complex that's involved in axon remodeling and in moving vesicles, trafficking vesicles around cells. Getting to the very end of the talk, and I probably run out of time, maybe a little, but obviously. We have the ability to sequence like you wouldn't believe. So Canada has invested $41 million in me and what I do, and what I've done is I've basically invested in high-performance computing. I've invested a little bit in infrastructure for sequencing, but more in bioinformatics and actually analyzing sequence, right? And so I can sequence like you wouldn't believe. And so we created a panel where we sequence all of the genes that are linked to associated with neurodegeneration on a single chip. And we can do this very, very fast. And I thought, well, so what would be a good population to apply this to? Where is there Parkinsonism and dementia in the world? Where is it prevalent? Where would it give us some answers? This is not just the genes for Parkinsonism and dementia that we know of, but it's also the genes for amyotrophy, for ALS, for motor neuron disease as well. I started off in the Faroe Islands, because in the Faroe Islands, Parkinsonism is far more prevalent. This is a group of islands in the middle of the North Atlantic. It's 19 islands in total, and they put their Parkinson's disease down to eating too much whale meat and too much puffin. You can get puffin in the Faroes, in the right time of year, and unfortunately I missed it when I was there, but puffins concentrate PCBs, polybichloorophenols in the food chain, and that's thought to increase the susceptibility to Parkinson's disease. But it's not true. The reason why is if you do some digging, and I did when I was there, this is a place called Tornavik. It's one of the villages, and it's up here. And this is another one down here in Sounder. I went to some of these villages, and basically what we find is that the prevalence of Parkinsonism is not just in the Faroes in general, it's focal. It's focal around very specific villages and families within those villages. This is an illustration of one of the families, and I'm now working with two very good, one an epidemiologist, an environmental epidemiologist, Maria Peterson, and Sarah Beck, who's a neurologist they're both Faroese originally, so they're both working Sarah works mostly in Denmark, but I've managed to raise the funds to get her to come home and see patients back on the Guerrero Islands. And so it's a part of a big project to actually sequence all 48,000 people on the Faroe Islands. And another place I thought I should come is Guam. Partly, I'm here, as I said, from a family, and I've had a really good welcome. I've been very much looked after and closely guarded by my nieces. And I've been having a really good time. This is me on the beach on Sunday, with Sophie and Katie, and armed to the teeth. make sure I don't get into mischief. And of course, many of you may know, Guam's been in the limelight for many, many years for a form of Parkinsonism and dementia and having an increased prevalence of that in some parts of the island. And it's been an enigma for a very long time. I was very lucky to bump into John Steele, I think, over the years. And John has very kindly made available to me not a lot of samples, because he doesn't have that many, but samples of Chamorro Islanders from Guam to try this little technique with. And it's only 100 samples, and only 71 of those samples actually have some form of neurodegeneration. The majority of healthy controls. One of the first things I did is looked in the genome of those individuals. They're all anonymized, it's all de-identified, well, more than de-identified, it's all anonymous to me. And the samples were taken, I think, many, many years ago. John can answer to this. Many years ago in the 90s, but and I realized many investigators have come to Guam over the years to do these types of studies, and I'm not one of them. And but I thought, let's try this technology, and sure enough we did. And first thing I noticed, this is Chamorro Islanders, this is Canadians. This is just a technique known as principal component analysis, where you look at the genome and say, like, where are people from? And sure enough, the samples that we had, because they've been in a liquid nitrogen bin for 20 years, I was a bit worried that people might have mislabeled them, muddled things up. So I wanted to make sure, first of all, they're really Chamorro. And I noticed that you can't really see it here very well, but there's 4 green spots here. These are my Canadians, and I'm thinking, what are they doing over in Chamorro gene pool? Very bizarre. Because it's very unlikely there'll be Chamorros in Canada, isn't it? Well, maybe not. They're actually Filipino. They're Filipino. And so one of the things, the resolution of the technique is not so great, because you can narrow a person down to a particular village, or maybe even a particular island, but more to a region, and probably a continent. So Philippines, the gene pool of Philippines here is very close, at least these Canadian Filipino individuals is very close to Chamorros. First thing I noticed. But it made me confident that what we had was actually Chamorros and not North American samples from Antigua species. And then we said about sequencing. And one of the things we found is that of the 71 individuals that were sequenced, we found that something like 10 of the 71 have gene mutations that have previously been described in the literature in other countries, in other parts of the world. The 3 of them, for instance here, have homozygous PINK1 mutations. I'll show you a bit more. 2 of them have dynactin mutation, which I didn't describe. It's a gene we mapped back in 2009. Also causes actually motor neuron disease in some families, causes frontotemporal amyloid dementia in some families, it causes Parkinsonism, Perry syndrome in some families. There's a novel mutation in families from Chamorro families here. We noticed a lot of variability, but not enough to cause disease, but a lot of variability in a gene called alsin, ALS2, which is a gene which is, if you inherit a mutation in a recessive manner, so 2 copies of this, or 2 mutations basically together in a recessive fashion, you'll develop motor neuron disease. And we noticed that this is quite frequent, at least in this very tiny sample of 71 individuals. So I was expecting to find maybe 1 or 2 known gene mutations when we ran our panel. Not 10%. It's far, far higher than I would imagine, and maybe it's just bias of sample. Who knows? Or somehow, everyone's come to see John, who has this genetic burden of disease, and this is the explanation. It's quite enigmatic. One of these individuals, and because John has done such a wonderful job, I think, over the many, many years of working with families and getting their trust and rapport, that he's managed to get brain autopsies from some people, some individuals, and they've been very carefully archived for decades. And so going back in the record with John's help, we managed to find, for instance, one of these individuals who's a PINK1 homozygote, who has this pathology. They have hippocampal tauopathy, they have amyloid plaques, that's rather like Alzheimer's disease in a sense. They have a lot of, this is the amygdala, a lot of Lewy pathology as well as tau pathology. This is a tau stain in the amygdala. This is the neuronal loss that you see in the nigra. So the nigra is wiped out. You normally see cells like this. There's no cells, it's just gliosis. And you also see some synuclein pathology, but mostly tau. It's very different. It's a cultural, you know that described as PINK1 families before, and the clinical phenotype, the clinical presentation is later, very, very different. It's a typical PINK1 disease, but nevertheless the mutation was first described in the Philippines, and actually the carrier frequency in the Philippines is about 8%. So you'd imagine that perhaps if some of the Chamorro ancestry is indeed Filipino, there would be a higher frequency of PINK1 disease on Guam, and sure enough, I think we just found that, for example. I'm going to stop here. There are many, many more genes for Parkinson's, and there's much more we can say and do about them. I wrote a review, it came out last year in Nature Reviews Neurology, of how this all works together. And this is a simple explanation, my layman's explanation. But I think it actually works better. It's like bits of a car, right? Now we know, where the battery is and the alternator and the radiator. We know the different components of the car engine. Right? We're starting to piece this together. We're starting to realize how this car is put together. We can start to work out how those different component parts work together to make that motor run. So this is my analogy for Parkinson's disease and genetics, to make it accessible to the layman. Lastly, the group, just to acknowledge the postdocs and medics in the group, and the graduate students in the group. But it's actually been, I really should thank the families more than anybody else, and the populations. It's really been a wonderful, wonderful journey for me around the globe, trying to solve this problem. And I think we're making a lot of progress. It's good news. Thanks very much. I'm happy to take questions. I've probably said enough anyway, but it's if you're interested. I'm going to be around for a little bit. So.
Can I comment, Matt, to say what a splendid presentation it's been. Left many of us way beyond untrue. But it clearly illustrates the advances now in the understanding of Parkinsonism and the genetic influence that is so important in its cause. And it makes us aware of other isolates of Parkinsonism and dementia that are quite similar to Guam. For many years, those of us who studied this disease on Guam have focused on the similarities with the disease in Japan, in the Key Peninsula, and in New Guinea, West New Guinea. And we thought these were exclusive, specific isolates of dementia and Parkinson's. But there may be others. And so each visitor, each foremost visitor, like yourself, for 70 years now, has brought new ideas, new thoughts to us on Guam about this very puzzling and enigmatic disease. It's a splendid presentation, and the opportunity to work with you and you colleagues in UBC to clarify the genetic contribution to Parkinsonism dementia on Guam is something that we really all look forward to. So we welcome you enthusiastically and your support and the knowledge that you're going to bring to us about this disease, which fortunately for Chamorros is disappearing. In other parts of the world, Parkinsonism dementia continues. It passes from parents to their children. But in Guam, that's ceased, apparently. So the children who had parents with Parkinsonism dementia no longer suffer. And I'm not quite certain how that reconciles with the genetic etiology, but this is the path we'll go down, I hope, in the years ahead. The heavy lifting, I think, is about to begin. So those are my remarks, but Dr. Chen is with us this evening. Dr. Chen has given most of his life to the studies of Parkinsonism dementia on Guam. We haven't always agreed, Dr. Chen and I, nor do we still, and still, we don't agree. But he has a long experience. And so maybe Dr. Chen would comment on the presentation and what thoughts it's brought to his mind.
Yes, I have worked on Parkinson's dementia country for about 40 years with the NIH, Neurosciences, and tried to look for genetics and some other environmental factors. Now, until 2005, these two diseases, ALS and PD, completely disappeared from Guam. In other words, for those Guamanians who were born after 1950, in normal cases, has a very dangerous. Meaning that I think the Guamanians have been healthier, their salaries, their good lives, so that could be the difference. But I think your presentation today, I'm very impressed that you went so deep into the molecular genetics, so deep, so many varieties of Parkinsonism, and I'm very impressed. Thank you very much.
Okay, on behalf of the University of Guam, I want to thank you for being our premier speaker and want to present you with this momentous
It's a rare medical condition, yeah.
Thank you very much, everybody. This concludes the lecture series for this evening. Have a good evening.